Hello and welcome to this lecture series about synaptic transmission. My name is Ranjita, and I have worked in the field of synaptic transmission since 2008. I'm currently leading a company called Brain Awareness GBG, which focuses on awareness and education about the brain and is located in Gothenburg, Sweden. Our brains contain 86 billion neurons. And each of these neurons have the ability or can have the ability to form thousands of contacts. And it is each of these contacts, individual contacts, that we are interested in. They're also called synapses. Neurotransmission is a spatially and temporally tightly regulated phenomenon. And it is interesting to think about how evolution has optimized the tightly regulated steps behind synaptic transmission in order to make our brains function optimally and to maintain normal functions like learning and memory. And perturbed synaptic transmission can lead to neurological disorders as varied as Parkinson's, Alzheimer's, epilepsy, and schizophrenia. In the next 20 lectures, we will be focusing on one question. How do neurons communicate with one another? In other words, we want to know the cellular and molecular basis of synaptic transmission. In this lecture, we are going to go through all of the steps, a little bit at a time, but in future lectures, we will be zooming in on each of these steps that underlie synaptic regulation. And we'll also spend time on the discovery behind the knowledge of synaptic transmission. Synapses are the places where neurons contact one another. And by the means of synaptic transmission, they communicate with one another. Synapses can either be electrical or chemical. Electrical synapses are fast synapses. They are made up of gap junctions, which in turn are made up of little channel proteins called connexons. And by the means of these channels, they pass current, but they can also pass metabolites. So gap junctions are important for electrical coupling as well as metabolic collaboration between neurons. Chemical synapses, on the other hand, are slow synapses. And they communicate by the means of small molecules called neurotransmitters. And these neurotransmitters bind to receptors on the postsynaptic neuron. They allow a little bit more flexibility and modulation compared to gap junctions. And chemical synapses will be our focus because they are the most common type of synapses. Chemical synaptic transmission can be roughly divided into these 8 steps, from the synthesis of neurotransmitters to the traveling of filled vesicles to the plasma membrane and to the termination of the signal. In today's lecture, I will go through each of these steps a little bit at a time. And in the future, we will focus on each step individually. The first step in neurotransmission is neurotransmitter synthesis and storage. But before we go into synthesis, we will acquaint ourselves with the most common neurotransmitters. And these can be divided into small molecule neurotransmitters as well as neuropeptides. In the following lecture, in lecture 3, we will talk about the most common synthesis pathways of these neurotransmitters. And neurotransmitters are made by the help of enzymes. They take precursor molecules and modify them, and then we get active neurotransmitters. Neurotransmitters are stored in vesicles. Vesicles are small membranous compartments, roughly 30 to 50 nanometers. And they are made in the ER. And they are transferred from the ER to the Golgi, where they are processed. And then they bud off from the Golgi to form independent compartments. They finally travel down by the help of cytoskeleton and motor protein down to the axon terminal. The final step is the filling of neurotransmitters into synaptic vesicles. And they're filled by the help of vesicular transporters, which make use of the proton gradient across the membrane, the vesicular membrane. And they pump neurotransmitters from the outside to the inside of the vesicle. So it is only once the vesicles have traveled down to the membrane, to the presynaptic membrane, that the action potential can perform its function on the vesicles. So in the next step, the action potential travels down to the presynaptic terminal. And in the following steps, this force will drive the neurotransmission. But before the action potential comes in, the neuron will occupy a more resting state. It will be at its resting membrane potential and be defined by its passive electrical properties. So the first question we ask ourselves is why is there voltage over the plasma membrane in the first place? As the action potential depolarizes the presynaptic membrane, voltage-gated calcium channels open up. Voltage-gated calcium channels can be found in N, P, Q, R, L, and T type. And they have different conductances as well as different voltage sensitivities. Once the voltage-gated calcium channel has opened up, it lets in calcium from the outside, from the extracellular environment, where the calcium concentration is about 1.5 millimolar, to the intracellular environment, where the concentration of calcium is only about 50 to 100 nanomolar. And this concentration difference in calcium is a major driving force for the opening of voltage-gated calcium channels. But there are many sources and sinks of calcium in the presynaptic terminal. Apart from the opening of the voltage-gated calcium channels, there is also influx from ryanodine receptors, which sit on the ER at calcium-induced calcium release. Also, IP3 receptors, which sit on the ER, open up and let calcium into the presynaptic terminal from the intracellular calcium store. On the ER, we also have Circa pumps, which pumps calcium back into the intracellular store, the ER. And another organellar buffer is mitochondrion, which can be found in the presynapse as well, close to the presynapse. In the plasma membrane we have plasma membrane calcium ATPases, as well as sodium calcium exchangers, which extrude the calcium from the intracellular environment to the extracellular environment. Another type of calcium sink are calcium binding proteins, for instance, calbindin. So it is clear that calcium regulation in the presynapse is multifaceted and it's very complex. But this has relevance for both vesicle recruitment to the membrane as well as fusion of the vesicle with the plasma membrane, which allows the neurotransmitters to diffuse out to the synaptic cleft and the postsynaptic neuron. So in the next step, we have the calcium, which has flown in through the voltage-gated calcium channels. It allows vesicle docking and neurotransmitter release. So when the calcium comes in, it causes the vesicle to go near the membrane and dock at the membrane. And this docking, this contact between the vesicular membrane and the plasma membrane is mediated by steric complex, a kind of anchor protein complex. So at the vesicle, we have the V snare, the synaptobrevin. And at the plasma membrane, we have T snares, the SNAP25, as well as the syntaxin. And when these V snares and T snares contact each other, there will be an attachment of the vesicle to the plasma membrane. But these snare proteins are not only important for vesicle docking and priming together with their attached factors, they're also important for the fusion of the vesicle to the plasma membrane, which lets out the neurotransmitters. And this fusion occurs when the fusion sensor, synaptotagmin in most fast synapses, when it binds to calcium. So once the fusion has occurred, the neurotransmitters will diffuse into the synaptic cleft. And this diffusion is rather fast because the synaptic cleft is only 20 to 40 nanometer large. And the concentration of neurotransmitter at the point of release can be up to millimolars. But not all the neurotransmitters are bound to postsynaptic receptors, but some of them need to be cleared by mechanisms that we will discuss shortly. So what happens in the next step is that the neurotransmitters, which have diffused over the synaptic cleft, will bind to receptors. And postsynaptic receptors can be of two different types. They can be ionotropic or metabotropic. But we'll start by talking about ion channels, which, when the neurotransmitter binds, open up channels and let in ions. And depending on what kind of neurotransmitter we have, what kind of receptor we have, the reaction will either be excitatory or inhibitory. For instance, glutamate is an excitatory neurotransmitter, GABA is an inhibitory neurotransmitter. But we can also have metabotropic receptors, and these activate downstream signaling pathways, and dopamine receptors are an example of metabotropic receptors. So in the postsynapse, we will have either an excitatory or inhibitory change in the potential. And finally, the signal has to be stopped. So the signal is stopped by removal of neurotransmitter, either by reuptake into the presynaptic neuron from which it just came, or glial uptake, where the neurotransmitter diffuses away from the synaptic cleft and is taken up by astrocytes, or enzymatic degradation. And one example of inhibition of presynaptic uptake are selective serotonin reuptake inhibitors, which are used in the case of depression. So there's one more step left. We have to take care of the presynaptic neuron. It has made a lot of vesicles fuse with this plasma membrane, and if we don't retrieve these parts of membrane, the cell will keep growing. So the presynaptic neuron uses mainly three different mechanisms to retrieve this membrane. And they're called kiss and run endocytosis, clathrin-mediated endocytosis, or bulk endocytosis. And depending on the situation, for instance, what kind of stimulus the cell gets, it chooses one of these pathways for endocytosis. So I have walked you through the main 8 steps for synaptic transmission. And in the next lecture, we will delve into detail and talk about neurotransmitters, about the different types of neurotransmitters there are. But one more thing. So this synaptic transmission that we have seen, this life cycle of the vesicle and of the neurotransmitters, it occurs recurrently in the brain, in the synapse, because presynaptic and postsynaptic neurons talk to each other by patterns of high frequency or middle frequency stimulation. And depending on the need and the experience of the synapse, it can either enhance the signal over time or decrease the signal over time. And it does this by changing one of the regulatory steps that we have just talked about. And this is called synaptic plasticity. And synaptic plasticity can take place on a short-term time scale as well as a long-term time scale. And this will be the topic of our discussion during our last lectures in this series. So thank you very much for listening. I know I've provided you with a lot of information, but don't worry if you didn't catch everything. We'll be going over the scheme of the 8 most important steps of neurotransmission many times over the next 2 years. Thank you.
Jättebra.
Tack.